Okay, good morning, and thank all of you for showing up promptly and alert to get the second day of our conference going. My name's Gilbert Whittemore. I'm currently the chair of the section of Science and Technology Law for the American Bar Association, one of the co-sponsors of the conference. And it's my pleasure and honor to be given the pleasant task of welcoming you this morning to the second day of what has been a very educational and productive conference, and I think we'll expect the same today. Just a little bit of information about what the entity is that I'm currently chairing, the section of Science and Technology Law at the American Bar Association. Officially, we're the section that is interested in looking at the interface of science and technology law issues. Unofficially, we're the section where all of the lawyers who are wannabe scientists tend to drift towards because they'd like to know about science without having to do all the really hard work. We try to be the section that looks at the issues that are just coming over the horizon or that are still not yet even beginning to rise. And we have members and we have committees that cover quite a broad range, ranging from information technology to biotechnology to outer space law to nanotechnology. And through all of those, obviously, our section has an interest generally in science and technology, technological research. And we're also very gratified that for a number of years we've had a liaison relationship with the American Association for the Advancement of Science, which leads us to at times be asked to participate or to co-sponsor in gatherings such as this, where you try and cross the professional boundaries and bring together lawyers and scientists in a setting that is outside of the courtroom. I really have just three basic points I wanted to make in terms of the welcome, the timeliness, the challenge, and then a thank you. I'm preaching to the choir a little bit by saying that this is a very timely conference. Just as two examples, the Chronicle of Higher Education within the last few weeks had a two-page article on research involving animals in university labs. And if you're like me and you follow the popular science press, you have probably heard in the last couple of weeks about the Lance Armstrong mouse that was an unexpected discovery in a genetic manipulation and ended up producing mice that apparently somewhat like Lance Armstrong have very good metabolism rates and long endurance and an unusual amount of strength, all due to manipulating or changing just one gene, which makes for great sound bites on the radio, but also illustrates that all of these types of developments we're talking about today are something that is now well above the horizon and is now becoming increasingly known to the public. The timeliness of a conference like this, I think, it illustrates another aspect of the rule of law that was talked about yesterday. Although you might not sense it if you watch the chase scenes on a lot of the shows on television that involve supposedly law and order, one of the major goals of any legal system is to try and take conflict and conflict that sometimes may be very deeply felt or run very deeply and try to find some way of responding to that in a nonviolent fashion. And an important aspect of trying to do that is not just having courtrooms and legislatures, but it's also having conferences like this where you bring people together, and as several of the speakers noted yesterday, where you try and move towards an area of exchange of ideas and dialogue without hiding the fact that there may be very deep differences of opinion or perspective. And I think that really is just to kind of put in context that what all of you are doing by participating in this today really is, I think, an essential part of our overall goals as a society of trying to achieve a rule of law. That leads to my second point, which is the challenge. It's always, not always, but it's usually fairly easy to get together a conference or a group of people who all agree. It's also fairly easy to get together a group of people, as in many continuing legal education conferences, people who are coming together because they want to learn how to do it. There's a new statute, there's a new regulation, this is how you comply with it, and everybody comes because they want to learn how to do it. And it's probably reasonably routine, if not easy, to get a group of scientists together who are basically saying, this is what I have done over the last year, this is my latest published research. They may have some pretty intense arguments about it, but they're all still on the same track. What's much more challenging is to try and bring together a conference like this one, which really is at the confluence of two major streams, one of complexity and one of controversy. And I think what really has worked well in the program so far, and I expect it will today, is we've seen the complexity being presented, both the scientific complexity, but at a level that those of us without PhDs can understand, and also the legal complexity. And also that, I think everyone in the room is aware, we're doing this with regard to an area of issues that are controversial, and yet I think the conference is doing a very good job of presenting various points of view in the controversy. That leads to the third and final point, which is really a thank you to those who put together and organized the conference, those who conceived of it, and those who put in many, many, many hours in planning the conference so that we are now seeing those of us like myself who are basically getting the benefit of being the audience without having been involved in the behind-the-scenes production effort, something that is so rewarding and easy to listen to because there are other people that put in a great deal of time to put it together. So I really would like to thank all of the organizers, and in particular the individuals that had worked so long and so hard to bring this together. And with that, like most lawyers, I'm capable of talking for 30, 40, 50 minutes. That's why appellate courts have traffic lights to tell lawyers when to shut up. What I'll now do is ask Deborah Runkle to come forward and introduce our first speaker of the program.
Well, thank you very much, Gil. And I am Deborah Runkle. For those of you who are here today for the first time, I'm from the American Association for the Advancement of Science, or AAAS, in Washington, D.C. And I am going to introduce our first speaker today. This first talk is really a continuation of the last panel that you heard yesterday, where people talked about the work, the scientific work they're actually doing with bioengineering and cloning technologies. Our speaker today, this morning, is working really more directly, I think, than yesterday's speakers to address real human diseases that cause millions of people suffering and very often death. Dr. Thomas Kaufman is chief of the nephrology division at Duke University Medical School. Nephrologists study kidney function and related diseases. And he's also a staff physician at the Durham Veterans Administration Medical Center. He was the principal investigator of the Mouse Models of Diabetic Complications Consortium group at Duke, which created new mouse models for diabetic kidney and heart disease. Dr. Kaufman's clinical interests include renal transplantation and hypertension, and his research includes the study of mechanisms of kidney injury in disease states and the role of the kidney in the regulation of blood pressure. His work uses molecular genetic technology to develop and refine mouse models of human diseases such as diabetic nephropathy, kidney transplant rejection, and hypertension. I give you Dr. Kaufman.
Thanks very much. I'm delighted to be here and very happy to talk to a group of lawyers where I don't have to mention the word malpractice. Nancy, who introduced me, came up and made sure that she knew the right definition for nephrology. And my mother-in-law actually doesn't know what a nephrologist is. We're not people who feel your head and by the shape of your head tell your fortune. And I'm not a urologist. That's what my mother confuses my mother-in-law. But actually, nephrologists are internists who take care of patients with kidney disease. So we take care of people who are on dialysis or have had kidney transplants. And I, along with my laboratory work, I see patients and really use that experience in the clinic or try to use that to inform what we do in the laboratory. And I'm going to use the other M word, not malpractice, but modeling during my talk. And really a major focus of our work is to try and use mice and use bioengineering techniques in mice to develop experimental paradigms that can help develop therapies, diagnoses, and treatments for the same patients that we see in the clinic. We'll try and sort of give you a slice of, in a very short time, give you a slice of kind of what we do. And Bill asked me to cover a little bit of the nuts and bolts of mouse modeling, which I don't think was covered in great detail yesterday. And I'm sorry, I was supposed to be here as part of this yesterday, but I was stuck down in Gatorland at University of Florida and had to take a tour of their athletic facilities and see all their basketball national championships in the last few years, which was painful, frankly. I tried to be polite, but what are you going to do? So in the first part of the talk, I'll just talk a little bit about some of the commonly used technologies for genetic engineering in mice, and then give a sort of broad strokes example using the diabetes complications as sort of a model of how we tried to use these productively. My other observation pertinent to this group, I think, is that, so I've been doing this work for more than 20 years, and the extent of protection that's evolved for mice during this period has really been phenomenal. And I think, back in the 80s, if you were doing experiments with mice, nobody cared, nobody looked, and now there's substantial protection, and there's a huge administrative structure here at our institution to make sure that regulations are being followed, animals are protected, and I think that's been a great advance for the field. So, I heard there was a problem with this area. So basically, I'm going to talk about the two sort of widely used approaches for doing bioengineering in mice. One is the sort of conventional and earliest technology for producing, generating transgenic mice, and then secondly, talk about the approach that we've used most frequently, which is so-called gene targeting, using homologous recombination in embryonic stem cells, which is another obviously loaded word and topic. So we'll start with the first one, the simple transgenesis, the so-called pronuclear injection, where one, again, using cutting and pasting of DNA sort of techniques on the bench top, generates a transgene for various purposes, and then that transgene is injected into the pronucleus of an embryo and incorporates randomly into the mouse chromosomes. And the constraints of the expression and the function of this transgene are really determined by the pieces of DNA that are put together in terms of the promoter and the protein for which the transgene is encoded. And compared to the more complicated technique of gene targeting, the techniques involved are relatively efficient, fewer steps, fewer constraints, and it's possible in many species, including mice, rats, pigs, even plants. And I think you probably got some examples of people talking about this in other species yesterday. This is the approach. So single cell embryos are harvested. The clone DNA is injected into the pronucleus of these embryos, and then it just like in vitro fertilization in people, this fertilized egg is implanted into a surrogate mother to generate transgenic offspring. This is the sort of technology. Here's an embryo. Here's the injection pipette. And here you can see that's injected right into the pronucleus. This little bubble here is the solution containing the transgenic DNA. And then these offspring of the surrogate mother are then small tail biopsies are taken. DNA is harvested, and then we do DNA fingerprinting again, just like we do for people to do DNA identification, and then can identify the presence of this transgene based on sort of simple molecular technologies. I'll give you an example of one. The use of this approach to develop or to address an important and problematic human disease, and that's the kidney disease that's associated with HIV infection, which has become, in incident areas, one of the major causes, most frequent causes of end-stage renal disease and dialysis. And in this case, the transgene, the piece of DNA that was inserted into the genome, was a fairly large portion, not complete portion, of actually the HIV genome. The key part of the DNA that's involved in propagating infection and transmission were deleted for obvious reasons. But this then truncated form of the HIV genome was inserted into the transgenic animals. You can see several different strains of these mice were generated. And this is the result. This is an animal that has the truncated version of HIV genome, and actually developed a kidney disease that very closely resembled that seen in patients who have this disease. So they develop proteinuria, which abnormalities in the filtration function of the kidney, and has been a very useful model in identifying not only the pathogenesis or how this disease occurs in humans. So it turns out that by using these series of animals, we were able to identify that, in fact, the reason people get kidney disease associated with this infection is due to the expression of gene products directly in various parts of the kidney, and has allowed us to focus therapies on that particular pathogenetic mechanism, and we've made real advances in preventing and ameliorating the course of this disease in human patients. Now, the second fairly common, or now very commonly used approach for bioengineering in mice is the so-called gene targeting, where here the goal is, and the opportunity is actually to very precisely manipulate the genome at a very specific region in the genome and make a very precise alteration. And there's great flexibility in the way the target genome can be manipulated. Most commonly, the target gene is inactivated. This is the so-called knockout approach. You've probably heard this jargon of knockout, the so-called knockout mouse. But there's lots of opportunities and flexibility in the particular alteration that can be done. One development that's happened really in the last year or so that I think is going to have substantial ethical and regulatory implications is now technologies have been developed where fairly large segments of human DNA can be inserted at precise locations in the mouse genome. Now, from the standpoint of understanding human disease, this provides the opportunity to generate a series of human genetic polymorphisms in a circumstance where their effects can be studied and isolated, and their contributions to human disease can be identified in a very powerful way. But again, these sort of approaches are at their earliest phases. The difference with this sort of approach is that the DNA integration occurs at a precisely determined location in the genome. As a more complicated technical process, and at this point, in terms of applications, is mostly limited to the mouse. And this, again, just to illustrate the scheme that it's involved. The key to this process are these cell populations here. They're embryonic stem cells, mouse embryonic stem cells, and you can see they form these little colonies, and they're actually derived from the inner cell mass of this single cell, or this blastocyst, which is an early mouse embryo, and you can see that the cells look very similar, and these cells have the almost amazing property, actually, of being able to be maintained in vivo for long periods of time in an undifferentiated state. And then when reintroduced into this milieu of the mouse embryo, then can resume their differentiation program and actually develop into these these chimeric animals here, which are made up with a combination of the cells, which can be genetically modified, and the recipient blastocyst. And these chimeric animals then, just like you see their coat color sort of patchy. These cells come from a white mouse. The blastocyst comes from a black mouse. That just like the coat color that's made up from constituents of both the ES cells and the blastocyst, that when this happens in the germline, then these animals can transmit the genetic modification through the genome as a stable genetic trait, and it provides a very powerful system for understanding the effects of these genetic manipulations on disease and physiology. This is just kind of a diagram of how that works, and some of the uses of this in biomedical research, obviously to understand the functions of a gene in vivo in the intact animal. So, for example, if our, as was mentioned earlier, we're interested in hypertension, which is a disease that affects 40 million people in the United States, has substantial associated cardiovascular mortality, morbidity. And in order to understand the contributions of a particular gene in the development of hypertension, one can't do that in a cell culture system, for example. One needs an intact cardiovascular system, blood vessels, and the ability to alter blood pressure in an intact organism. The key use, but to understand the role of genes in development, as I'll talk about, we can use these approaches to try and develop models of human diseases that can be used for therapeutic diagnostic purposes, and to try and understand causative connections between naturally occurring mutations and diseases and disease phenotype. Now, the power of this approach has recently been recognized nationally. So our neighbor and my friend Oliver Smithies, who actually was responsible for developing the technology associated with gene targeting, was awarded the Nobel Prize for Medicine and Physiology just this past year. And we can say, University of North Carolina here, right, in this conference, it's okay to mention that, okay, right. And actually, I personally spent a year on sabbatical early in the 90s with Oliver, who's really a great human being, has made substantial advances that have helped understand a number of different human diseases, including cystic fibrosis, thalassemias, also hypertension, atherosclerosis, very powerful insights into understanding how these diseases work in people, and to help us develop new approaches to manage and treat these diseases. 5 minutes. So then I'm going to briefly then just mention, it was alluded to earlier, our work in diabetes. So as a nephrologist, diabetes is sort of the scourge of my patient population. So, and this actually, for those the uninitiated, this is a glomerulus, which is the filtering unit of the kidney. So this is the subcellular organelle that allows the kidney to remove waste products from the body. And in the United States today, diabetes and the complications of diabetes are the leading cause of end-stage kidney disease in the United States. So about 50% of our patients who are on dialysis, when I round in my dialysis unit, are there because of this disease. And it's last year we spent about $12 billion with a B dollars on dialysis care, and this is a major driver of that expenditure and that human suffering. And so we've been interested, obviously, in trying to figure out how to not only prevent our patients from getting this, but once they have it, to develop better treatments once they have the disease. And with the forecasted expansion and projected expansion in sort of overall body mass and the obesity epidemic that's predicted in the next 10 years, and the associated diabetes that goes along with that, this disease worldwide will be a substantial problem. And one of our issues is that we don't have good models. I'll say that again, but that really is, I mean, these are required to understand how these diseases are caused and how to treat them, that recapitulate this disease. And so we've been a part of an NIH-sponsored consortium to improve our ability to study this disease, and we've used combinations of these technologies to address this. And I have 1 minute left, so I'll say, and we've made some progress, and we have more to go. But let me, I'm getting signals up front, so let me stop here, and I'd be glad to answer questions or have some discussion. Thank you for your attention. Oh, okay. Okay, sure. So I have a question about protection. So I mentioned that protection of mice has improved over the last 20 years. What was it like 20 years ago? What protections are there now? What do you attribute the change to? Well, I think as I mentioned, 20 years ago, mice were really not on the radar screen of any of our regulatory apparatus. Certainly, I think nationally, so really very little regulation in the 80s to somebody doing mouse experiments. And I think a big step in this was the Animal Welfare Act, and also because of the power of these approaches, the number of people who were turning to the mouse in biomedical research. And so today, to do the sorts of experiments that I'm talking to you about today, from the investigator standpoint, we have to, I mean, we have the oversight is almost, I mean, virtually identical to the oversight that one would have to do experiments involving humans. So there are, for me personally, there are two institutional animal care and use committees that all of my protocols are vetted through. These committees, as you probably know, are made up with a combination of scientists, veterinarians, lay people from the community. They're very rigorously evaluated, and in fact, in the past 5 years, useful and systematic protocols for overseeing housing and analgesia and all the important things that need to be done for animal protection have been put in place. So in my office, my laboratory, we typically spend maybe 30% of our time, our research time, related to regulatory welfare and oversight issues. So it's a very robust, appropriately robust system, and that mirrors almost in every way the same sort of oversight that we have for research involving humans. Next question is, do transgenes receive genetic material from the pronucleus of the host embryo, or is the host more like a surrogate mother? So the genetic material actually comes from cloned DNA that can come from any source, really. So it can come cloned from human DNA, for example, often pasted together from various sources. So let's say that you want to, or you need, or you'd like to have your transgene expressed in the kidney. Well, there are certain promoter regulatory DNA elements that may have been identified that will drive expression in the appropriate area, and that can be pasted together with whatever your transgene is. So the transgene is all from exogenous DNA, and through sort of mysterious mechanisms that we don't understand, it gets incorporated into the chromosomes of the embryo, which is then implanted through this in vitro implantation process. So I hope I answered the question. So a couple of questions. What percentage of transgenic animals express the genes expressing the genes that are the intended outcome. It's variable. So that's one of the especially of the pronuclear transgene process, is it's relatively random, and just because the transgene gets incorporated into the chromosome doesn't mean that it's going to be expressed. It needs to go into a place where the DNA, I mean, as we found out later, a lot of this was done kind of in the dark, but now it needs to insert into an area of the chromosome that's structurally receptive to the transcriptional apparatus. And again, that's one of the advantages of the targeting approach, because you can really precisely know exactly where the exogenous DNA is going, and you can do a lot of actually in vitro testing ahead of time to be certain that the things work the way that you hope they will, that were required for your experiment. What policies and procedures are in place for handling the failures? The guide appears to defer to standards set by the AVMA, which in turn seems to defer to researchers. What is the reality? So, I mean, there are standard procedures for euthanasia and that for animals where these fail, and or reusing animals for other purposes. And we have very rigid guides in place in our institutional regulations for how these are handled. So this kind of stealth question, but if people have comments or questions or complaints about what I'm saying, pipe in. I've raised a bunch of sons, so I have 3 boys, and so I'm used to getting yelled at, and I'd be happy to do that.
I don't think I'm going to yell at you, Dr.
Okay.
Kaufman, but rather thank you for your very...
Are you going to yell at me if I don't step down now? All right, get out of the way, right?
I came with the hook.
Okay,
So
all right.
thank you very much.
All right.
Join me in thanking you.
All right, thank you.